Hello, everyone. Can you hear me? Michael, it's okay? Okay, great. So I'm Pierre Brosset. Will be sharing this session with Michael Roussel in Rennes. I'm professor of pathology in Toulouse, and it's my pleasure today to introduce the speaker of this Redtalk, who is Professor Olivier Elemento. Olivier is professor of physiology and biophysics and director of the Carrill and Israel Englander Institute for Precision Medicine at the Weill Cornell Institute in New York. This institute is a large multidisciplinary institute that uses precision medicine technologies and informatics to uncover the molecular mechanisms of disease and individualize disease treatment and prevention. Olivier is also Associate Director of the Institute for Computational Medicine, Director of the Laboratory of Cancer System Biology, co-leader of the Genetics, Epigenetics, and System Biology program in the Meyer Cancer Center at Weill Cornell Medicine Institute. He was recently appointed Associate Program Director of Weill Cornell Medicine's for Clinical and Translational Science. Olivier Elemento received his bachelor's degree in mechanical engineering from University Paul Sabatier in Toulouse, France, a master's degree in mechanical engineering in the INSA Toulouse, an school of engineering, a master's degree in intelligent system from the University of Paris, and doctorate in computational biology from the University of Montpellier and the CNRS in Montpellier Institute of Human Genetics. And so it's a great pleasure to hear Olivier, and I would like to say that his scientific production is very impressive, which up to 400 papers in high-ranking journals. So thanks, Olivier, for participating to this meeting. And so it's your turn now to present the results of your work.
Yeah, thank you so much. I really appreciate. Thank you so much for inviting me to give a talk. I'm really happy to be here today and happy to tell you about our work. So the title of the talk is going to be Single Cell Pathology Landscape of Diffuse Large B-cell Lymphoma. My lab has been working on diffuse large B-cell lymphoma for a long time. What we do here at Weill Cornell is really to specialize in the analysis of clinical samples with the idea of using the information that we get from clinical samples to personalize medicine. I should say for a long time, we've been focusing on the tumor cells and the mutations that drive them. And I would say for a long time, we've not really paid too much attention to the tumor microenvironment. And in the field of lymphoma, as you will see in the next few slides, I think there's been a bit of work in that space, but not enough. And I think we're really uncovering a lot of really interesting biology, through the analysis that I'm going to tell you about. I think we need to keep that in mind, right? A typical tumor, whether it's a solid tumor, lymphoma, or other types of tumors, is not just a large number of tumor cells. It's really a combination of tumor cells and many, many other cells that are not tumor cells. That includes immune cells, and there are all kinds of immune cells in tumors. That includes blood vessels and also other types of cells such as fibroblasts. So that's really the way that tumors look like in patients, in people. And I think we can't really ignore, this complex ecosystem, because we know now that there are lots of interactions between the tumor cells, the immune cells, the fibroblasts, and essentially the tumor cells are really directing the microenvironment to achieve better sort of phenotypes and sort of improve their own niche. So this complex ecosystem is really, really critical. It's been difficult to study this ecosystem for many years because we didn't really have the right technologies. We have done in our lab a lot of profiling of tumors, a lot of bulk profiling. So we take millions and millions of cells and we extract the DNA of all these cells at the same time, or the RNA of these cells, and then we sequence the DNA, or measure the expression of all genes within these tumors, and we lose completely the ability to kind of say where different genes are expressed, or we even lose the ability to kind of reconstruct the different tumor cell populations. We really lose this information that I think is really critical. So in the past several years, here at the Institute, we've been focusing on how to recover, reconstruct, discover all these complex structure, and that's basically going to be the focus of the talk today. I do want to say that there's been a little bit of work in lymphoma on the microenvironment. This paper actually came out a few years ago in 2008 from the Stout group, looking at expression profiles. It was done using microarrays back then, but they realized that there are some stromal signatures, basically genes that we know can only be expressed in the microenvironment, for example, in fibroblasts or in immune cells, that according to this analysis did correlate with survival. So it kind of implied maybe an important sort of correlation with the biology of a disease. I should say, this paper really, I think, was highly cited, but I have not seen a ton of follow-up on this paper up until a couple of years ago. So actually, another group here at Cornell Medicine, led by Leandro Cacchetti and with Ari Melnick and John Leonard and Giorgio Ingirami, actually was able to put together a very large collection of DLBCL RNA-seq profiles, expression profiles, and run an analysis that is basically sort of very, very similar to what we could call deconvolution. So the idea is, as I said, when you analyze many cells at the same time, you essentially get an average expression signature of the many cells, right? But that there are some cells in there that are different and coming from different cell types. And that these cell types express very specific genes. Like we know exactly what are the genes that are specific to, let's say, T cells or B cells or macrophages, even M2 versus M1 macrophages. All of these cell types have very specific expression signatures. So what you can do if you have expression profiles assessed using bulk methods is to ask what is the expression levels of those genes that are unique to a particular cell type, right? And if the expression level is high, what it means is that you have a lot of this cell type, because these genes are only expressed in this particular cell type. So this is the idea of deconvolution, and it's been reasonably powerful, especially in being able to assess basically samples that have been profiled in the past and that you can kind of put together into a larger database and that you can reanalyze. But it comes with lots of complications, including the fact that we're not always sure what are the signatures of some cell types. There's still some cell types or subpopulations that don't really have a very clear signature. It's hard to actually really deconvolve samples into very detailed, sort of cell types. And obviously, I think a key aspect of the limitation of this approach is that you lose the spatial structure. Like you don't know whether you have tumor cells in proximity of, let's say, certain types of T cells or myeloid cells. You completely lose the spatial architecture of the tissue. Despite all this, this was a really interesting analysis. And in this analysis, they were able to identify 4 different types of lymphomas based on the expression signatures of these different cell types. A GC-like, a mesenchymal sort of subgroup, an inflammatory subgroup, and a depleted subgroup, an immune depleted, so like an immune cold subgroup. So that was, I think, a great starting point. But obviously, as I said, we wanted to really understand the microenvironment of these tumors in a lot more detail. Here at Cornell Medicine, as I said, we really are interested in this problem in general. We have invested in technologies that allow you to understand the microenvironment or the spatial architecture of tumor at single cell resolution. The one that we really like is this one in the middle. It's called imaging mass cytometry. Some of you may have heard about CyTOF, which is basically a single cell proteomics method that uses antibodies as a way to quantify the amount of particular proteins within individual cells. As it turns out, you can do cytoph on tissue, and that's really what this technology is about. I should say again that the other technologies, some of you may be familiar with Codex. Some of you may have heard about some of the spatial transcriptomics technology that are popular, but have also a downside, which is they're not yet single cell resolution. So that's why we like this imaging cytoph technology, because it is really single cell resolution, as I will show you. And it gives you really detailed maps of the tissue, and I think reveals a lot of really interesting information. And I think what I love about it is that it allows you to kind of watch tissue, watch cells in action. I always say, I think it's really amazing to be able to almost like see the tumor cells and the immune cells and the different cells. It's almost like watching a battlefield, where you can see the soldiers, kind of battling each other or helping each other. I think this is, really, really important and pretty profound, I think, in terms of what we can learn about the disease. So just a few words about imaging mass cytometry. So this is the way it works. So you start with tissue sections that you stain with multiple antibodies, just like the cytoph. The antibodies are labeled with different metals, and the weight of a metal is very unique. So each metal has a very unique mass, and therefore, we're able to detect the abundance of the different metals, based on the fact that they have a unique mass. So once you have stained the tissue sections with antibodies, what you do is you put the stain slide into a device that has a laser, and what the laser does is to ablate, vaporize, if you want, the surface of a tissue, and the vaporized ions and the metals, basically ionized metals, as a result of the laser ablation, go into the cytoph for quantification. And so what you actually measure is the abundance of the metals across the different the tissue section, and the abundance of the metals is correlated with the abundance of the markers that the antibodies recognize, right? And so the nice thing about using metals is that there's no interference between the metals. There's not like fluorescence, where there's interference between different tags. The metals, basically don't interfere with each other. So you can sweep in and out markers, and it's very powerful as a result to expand a panel or try a new panel. The limitation of this technique here, unlike spatial transcriptomics, is that you can only look at up to 40 markers at the same time, 40. So this is not a technology where you have ability to look at many hundreds of thousands of genes at the same time. You're limited. It's a limitation. I think it can also be seen as a strength, in the sense that it kind of forces you to think about what you want to interrogate, what are the hypotheses that you want to ask, from your tissue, and I think that's powerful in that sense. But what comes out of this analysis here is essentially multiple maps of the abundance of the markers across the same piece of tissue. Each marker has a different map, basically, and then we kind of put everything together. And then what we have to do is basically image analysis. The problem of analyzing this kind of data is image analysis. For some of you who may have worked with sequencing, this has nothing to do with sequencing. This is a non-sequencing approach to the analysis of tissue. This is what a machine looks like here. If you were to see behind this Hyperion big box here, that would actually be a cytoph. So the Hyperion kind of feeds into the cytoph. As I said, the ionized metals go into the cytoph. One fun fact is that the laser that's used to ablate the surface of the tissue, it heats up the tissue at the same temperature as the temperature on the surface of the sun. So these are very hot lasers, as you can imagine, which explains why you have these big pipes, here that we use to cool down the laser, because otherwise the machine gets very, very hot. But either way, I think this is really a very powerful technology. It requires having a dedicated technician. Hiran, who's here, is a dedicated technician. She operates the two cytophs and the two Hyperion, and it's critical to have somebody who's working in this full time. This is not something that you can do in your lab, kind of on a bench, with rotation students. This requires a lot of dedicated time and work. I'm hearing the chat. I can't see the chat. I'm hoping that everybody can hear me. Juliet has my text, my cell phone number. So please, Juliet, let me know if there's any technical issues. So this here is just to show you what the tissue, what kind of tissue that goes into the machine looks like. So we can either do whole slide analysis, which is what you would see here. But what we actually like to do more, because of the ability to improve and expand the throughput, is actually to use tissue microarrays. So tissue microarrays basically kind of the slide on which you put multiple calls from multiple patients. You can see this here on the right side. Each dot here, or each little dot here, is one patient, right? So in this slide here, we can basically combine multiple samples from multiple patients. And we are able to analyze multiple patients at the same time, using this technology. That's one downside of this technology, is that it's also a bit slow. So the laser is kind of moving on the slide and trying to ablate as quickly as possible. But it's not fast enough that you can really ablate a tremendous amount of surface. So we have to compromise a little bit and agree that we prefer to look at hundreds of patient samples, if you want, as opposed to look at individual patients, with a very, very broad area of analysis. Okay. So as I said, my lab has been working on diffuse large B-cell lymphomas for a long time. I think one thing that's really interesting and important about diffuse large B-cell lymphoma is that they are pretty much always treated with about the same combination of drugs. The combination is called R-CHOP, and I'm sure many of you know this. Combination of 5 drugs, and these combinations are obviously quite effective. Quite a few patients are basically functionally cured as a result of this kind of drug combination. It's actually, we do work quite a bit on drug combinations in my group as well, and use things like machine learning as a way to predict combinations that are effective. I'd say that lymphoma is, I think, a great example about the impact of using combinations of different drugs, to really kind of impact the disease, I think, in a very positive way. But as with every cancer, some patients respond really well. Let me just drink a bit of water. And some patients don't respond as well. So there are responders, non-responders, and obviously we always want to understand, predict ahead of time based on diagnosis biopsies whether a patient is likely to respond. As I say, traditionally this has been looked at mostly focusing on the cancer cells, but I'm hoping to convince you that there is very precious information in the microenvironment that is connected, correlates with response, I think, in ways that we need to really investigate more, because I think it's really, really interesting and fascinating. So thankfully for this kind of disease here, we had access to a very large cohort of patients. And the credit here to this really goes to our colleagues in Vancouver, David Scott, Christian Steidel, at BC Cancer. I mean, they've really been pioneers in collecting samples from hundreds of patients, annotating the samples really well, doing all kinds of multi-omic profiling. They built, I think, really an amazing resource that is really fueling investigations in these kind of lymphomas, across many different labs, and they're really wonderful collaborators. This project is also a joint project with Biomedicine, who's also a well-known B-cell lymphoma specialist, here at Cornell. The two main drivers of this project is Dylan, who's a grad student in my lab. He's co-advised with Ari, and Hiran, who I already mentioned, who's the technician who's operating the technology and is really amazing at doing that. So that the core that we had is really amazing. It has 400 DLBCLs, 1 to 2 punches per tumor. So some of these little spot here sometimes may come from the same patients. You can have 2 punches from the same patient, and obviously that's important to get a sense whether different areas of the tumor behave in a similar way or not. I don't actually have slides on this. We can talk about this during the question phase. But by and large, I mean, what I'll say ahead of time is that we actually see a lot of concordance in terms of the microenvironment and the tumor cells between two punches located in different locations of the tumor when we have two punches. Obviously, those two punches are selected by pathologists. There's a bit of bias towards areas of the slides that are rich in tumor cells, for example, but I think the homogeneity, I think, is substantial across different locations as far as we can tell. So we image 400 DLBCLs. This is a very data-rich type of technology, as you'll see. This gives us in total about 7 million individual cells. As you'll see, this is beautiful data, single cell resolution, 7 million cells in total across this cohort. So that's, a large data set. But the beauty of this cohort, as I said, is that we had also bulk RNA-seq, so that we can look at expression profiles, maybe of the entire biopsies. We have mutation data, copy number alteration data, and we have clinical data, which is, obviously the most important, the ability to, obviously follow patients in time and assess the response to chemotherapy and correlate that with all of the findings of this study. So as I mentioned to you guys, I think the critical aspect of this technology is the choice of the markers to go after. We can only choose 40 at a time. We can, stay in 40. And the 40, by the way, just comes from the fact that we have only about 40 different metals that can be used with this technology. We conjugate the metals to the antibodies ourselves. There are only 40 metals that we can use in this technology. So, that's the limitation. If tomorrow somebody, finds a 41st, metal, then we can have 41 markers. That's the major limitation. It's not fundamentally technical. It's really based on the availability of the right metals. I told you that you can do 40 at the same time, and here you can see that we image 64 markers. So it's not 18, right? So I think the reason for this is that we actually are working with panels of 40 markers, but to be able to kind of map the panels into each other, there are markers that are shared between the panels. So that's why we have 64 markers in total. The type of markers that we chose here, as I said, depend on how you want to interrogate tissue. So we have a lot of immune markers to be able to track where you have myeloid cells, different types of T cells, the B cells, other types of immune cells. We have functional markers that we can use to assess the function and the phenotypes of individual cells, whether they express specific checkpoints, the metabolic states of some of these cells, whether they proliferate, with Ki-67, whether they have expression of specific transcription factors. We can actually even look at histone-modifications using this. That's the beauty of this technique, right? It's using antibodies. So as long as you're able to raise antibodies against a specific marker, you can use it with this technology. So it's we have these additional markers here. We have transcription factors, p53, markers of apoptosis, and so on, MHC, and so on. And we obviously have markers that are a bit more kind of structural. For example, to be able to track where you have blood vessels, lymphatic vessels, extracellular matrix, which is not something that you basically visualize within the cells. It's kind of outside the cells, as well as other things like vimentin or H3. So the technique requires some level of optimization, which we do typically using normal tissue. Part of the reason is because typically with normal tissue, for example, lymph nodes in this particular case here, we know what to expect in terms of the cell types that are supposed to be there, the abundance of some markers, so that kind of allows us to, we use them as positive controls, if you want. Allows us to titrate the amount of antibodies, and so on. But it also gives you a sense of what the data looks like, right? Data is really beautiful, and the colors, by the way, so now it's real colors, right? Every image is black and white, but we can use fake colors as a way to kind of highlight different multiple markers at the same time. What you see here is that we achieve single cell resolution, in fact subcellular resolution. You have clearly you can see cells that proliferate, express Ki-67, CD11C expressing cells, so basically macrophages, here with the nucleus, that if you had a nuclear marker, you would see the DNA, here. You have CD8 T cells, the periphery. So, it gives you a sense of what the data looks like. So that was normal tissue, and this is what we get using tumor tissue from some of those DRBCR cores. You see the cores here. That's sort of maps to the slide that I was showing you with individual with multiple cores. This is showing you some of these cores here. This is showing the raw data. So you can see the different signals, right? I mean, you're able to see the cells, right? You're able to see also things like some level of structure, such as a blood vessel. This is a blood vessel that was cut across, right? So you can see the elongated feature of a blood vessel. So there's a ton of information about the different markers as shown in this slide. So as I said, the analysis of these data is basically cell segmentation. It's computer vision, right? You have to find the cells. It's image analysis. You have to find the cells. You quantify the signal within the cell. You can actually look at subcellular resolution. So you can not only look at the entire cells, but you can find the nuclei. You can quantify the amount of protein expression at the membrane, within the nucleus, in the cytoplasm, and so on. It's not, the resolution is not amazing in the sense that you can't, for example, look at mitochondria, like you wouldn't really see them well, but you can resolve some of the major components of the cell, for sure the membrane, the cytoplasm, and the nucleus, very, very easily. And you can also look at other things, such as the shape of the cells, the morphology of some of the cells, whether they're elongated cells or polarized. You can look at M2, M1 macrophages and look at polarization, for example, which is something that we do. Not shown here, but this is another aspect of this data, is that you get, for all 7 million cells, you also get information about the shape of the cells. So the markers the basic markers that we've been using to, for example, to address or to assess individual cell types, then we can use them to say that individual cells belong to a particular cell type, right? So if you look, for example, for cells that express CD20, that's a very marker that's very specific to B cells. So that basically tells you what are the B cells among the 7 million cells. A subset of them express CD20, and so they are basically the B cells. And you can do this for all of the basic markers like CD8 and CD4 and so on. So that gives you a very high-level assessment of the cell types within your tissue samples. But what you can do is to go into more details, right? Because you have many more markers, so you can actually subdivide your cells into cell populations. This is done using essentially kind of unsupervised clustering, right? Imagine that we basically look for population among the 7 million cells. We look for populations of cells that express similar markers, and we group them together if they express very similar markers. When we do this, we find some really interesting subpopulations. So, for example, we find that there are, across our 400 DRBCLs, there are 6 different subtypes of B cells, and we'll get back to this. I think there's a lot of really interesting biology there. But we wanted to focus on the microenvironment, so we actually were able to subdivide the population of cells in the microenvironment into about 24 or so different cell populations. You have about maybe 15 or so myeloid cell populations expressing different combinations of myeloid markers. Some of them express interesting markers such as IDO or immunosuppressive markers, kind of making them like a little bit like M2 macrophages. You have granzyme B expressing myeloid cells. You have obviously lots of T cells. We have some nice T cell markers, and you can see, for example, there are CD8 T cells, CD8 T cells that express PD1s, CD8 T cells that express PD1, and granzyme B and other combinations. So very, very detailed analysis of the microenvironment. And so the beauty of this is that now you can go from the picture on the left, which is the raw data, right, where you can't really tell easily what's what, to a much more simplified picture on the right side. Same core, but now we found in each of a cell, and we essentially are able to name the cells, right? So the green cells are the B cells in this picture here, because they express B cell markers. The red cells are the CD4 T cells, because these are the cells that express CD4 as well as additional markers, and so on. So you can see the diversity of cell types that you have within a tumor. And I find this kind of image, really amazing, because it really shows you the complexity of a tumor, right? You would think that, in some cases, you don't really know what to expect, right? But You think that there will be compartments. You think that there will be really interesting structures. And, I mean, there are such compartments, but they may be a bit more subtle than one would expect. One thing that you can see here, if you look at this image, maybe long enough, you can see that on the upper left, here, is actually a compartment that is very B-cell rich. And on the upper right and the bottom left, this compartment here seems to be much less B-cell rich and maybe has more CD4 T cells. So you can see, there's already some level of structure that you can assess using this, basically this technique, and even within small cohorts like this. So now that we have this information across 400 different tumors, right, we can do all kinds of analysis. We wanted to really run this analysis focusing on the microenvironment. So one analysis that we did was to try to ask whether, if you were to count the number of cells in the microenvironment, which is what is being shown here, per tumor, right, and then you cluster the tumors based on these counts, based on the abundance of cell types within the microenvironment, are you able to find structure? This is essentially more or less ignoring the B-cell compartments for a second, focusing on how the microenvironment can potentially drive the clustering of the tumors. And what's really amazing, right, is that we didn't really have too many expectations when we did this, but we can see very clearly here that what emerges from this analysis is a subdivision of the tumors into 5 different subtypes of microenvironment. There's one subtype that the paper I showed you at the beginning already had found, and I think has been known for some time, which is a cold microenvironment. Tumor cells, rich in tumor cells, but overall very few immune cells and stromal cells. Basically very cold tumors. But what is really interesting is that we also find the opposite. There's actually a subtype of microenvironment that we call cytotoxic for a variety of different reasons. But if you look carefully, it basically consists of multiple myeloid cells, a very large number of myeloid cells, as well as T cells that express some really interesting markers. A lot of them express immunosuppressive markers for macrophages, but also express, for example, PD-1 expressing T cells with coenzyme B, which is essentially why we call them cytotoxic. We call this subtype cytotoxic, but also essentially these are exhausted T cells, right? So, this is an immune-rich microenvironment that, in some ways, consists of lots of immune cells that I would say are not very functional. It's a dysfunctional microenvironment, but still extremely immune-rich. You also have intermediate states, CD4 rich with lots of CD4 T cells, like you see here, including some of them expressing PD-1. You also have an inflammatory microenvironment, which is a bit more, I think, more like a normal lymph nodes sort of type of microenvironment. And you have, something that is a bit different, like a stromal-rich microenvironment with polyplanin and some other CD31 and so on that are a bit more basically rich in terms of blood vessels and lymphatic vessels. So, as I said, we were surprised, but, we did see that there are 5 different subtypes of microenvironment in this data. And, obviously you want to convince yourself that these subtypes are real, right? So, there's nothing better than just looking at the data. I always tell my students, look at the data, convince yourself that what you found using an analysis is real. There's nothing better than that. And, it's actually not so hard to be convinced, right? These cold tumors indeed are very cold, very green, only B cells. These cytotoxic tumors here have a lot of these purple cells, that you see here, these macrophages. They also have a lot of T cells and different types of T cells, right? So they're really cytotoxic in that sense. And you have CD4 rich tumors, with some of the red cells and some stromal cells, sort of tumors, with more of these lymphatic cells you can see here. So I think that this is really going to be convincing. This is looking at 3 different examples of tumors falling into within these subtypes. But at the end of the day, right, you can say, well, that's great. You found 5 different subtypes of microenvironment. Does it matter? Well, it turns out that if you look at survival of the patients that fall into these different subtypes, that seems to matter. Actually, the poor performers, the patients who don't do well in terms of chemotherapy response, actually turn out to be patients that fall within this the cytotoxic group here. These are the patients who really do the most poorly when it comes to response, both in terms of overall survival, but also in terms of progression-free survival. Actually, in terms of progression-free survival, and maybe there's a trend also in overall survival, the second worst performer in terms of chemotherapy response, actually the cold tumors. I mean, to me, this is fascinating, right? Because you have these two very distinct, very different subtypes of microenvironment. One that is super immune rich, another one that's super immune poor. They actually do, almost equally poorly. To me, what it really means is that there has to be some kind of synergy between the chemotherapy and the microenvironment. And more or less, I think what you need to do well as a patient with lymphoma, with this kind of lymphoma, is almost a reasonably healthy microenvironment, with enough immune cells, so that basically the immune cells maybe can facilitate the work of the chemotherapy or synergize with the chemotherapy and eradicate the tumors. I think this is, to me, I think one of the explanations. Now, how exactly that happens, I don't think anybody knows, and this is something that we do want to study moving forward. But moving back a little bit, to the tumor cell populations, because I think there are some really interesting patterns as well. What we're able to do within those 6 tumor cell populations I was telling you about is to label them as basically based on cell of origin. Likewise, if you've been working in this field of B-cell lymphomas, you know that there's been a classification of tumors, the ABC and GCB. ABC stands for activated B cells, GCB is germinal center B cell-like. These are essentially cell of origin sort of differences in terms of the tumor cells and where they come from. So traditionally, the tumors have been labeled as ABC or GCB. What we see when we look at those 6 different B cell populations, and by the way, these are tumors, right? So these B cells are by and large, mostly 95% of them are tumor cells. We know that. There's maybe a little bit of normal B cells, in there, but very few. But what's really interesting is that, we can subdivide those 6 populations essentially into ABC or GCB subpopulations based on the markers that they express. There are markers that tell you whether a cell is ABC or GCB. And all of these different tumor cell populations, fall within either ABC or GCB, 3 of each. But now what's really cool is that, we can go back to individual tumors and look at the composition of these tumors in terms of only the tumor cells. We can actually completely ignore the microenvironment in this particular analysis and count how many tumor cells of each of the 6 subtypes you have. And what's really interesting is that, you see quite clearly that every tumor is in fact a mix of multiple combinations of tumor cell populations. There's always a trend, right? So, you have these tumors here in the middle. In fact, these are the ABC tumors. The ABC is labeled using a different technique that is used to label the entire sample. But obviously, those are enriched in tumor cell populations that we label as, ABC, as you would expect. But even those actually do have, at least for some of them, some levels of some of these blue subpopulations that are GCB-like. So every tumor is in fact a combination, but there are trends. There are some tumors that are more dominated by either the GCB or the ABC populations. But I think it's clear that every tumor is in fact a combination. And you can see that, if you actually look at the data and look at the markers that define ABC or GCB, you see that within the same tumor, you basically see those cell populations quite often coexist. So I think what we've been doing, and I think in some ways it kind of makes sense, right? But we've been calling the entire tumor ABC or GCB. But I think every tumor is in fact a combination of different components of different subpopulations. And the average obviously leans one way or another, but in fact it's more complicated than just saying that everything is ABC or GCB. We can in fact look at the correlation between some of these tumor cell populations and the GCB and ABC subtypes, and we see obviously that there is a strong correlation. Now, what's interesting is that we believe in this classification based on the microenvironment, and we ask whether there's a correlation between the microenvironment and this kind of classical ABC GCB subtypes, and it turns out the correlation is actually not very strong. the microenvironment, there's some level of correlation. For example, the cold tumors, maybe a bit more, ABC than, expected by chance, but the correlation is actually pretty minor. So, I think we have a lot to learn about, the way by which the tumor cells, relate to the microenvironment. I'll continue on this theme. And likewise, we can look at specific markers. This is kind of an expansion of this analysis. We can look at specific markers. And, I think what's nice about this analysis here is because we can really focus on the tumor cells only. We can assess the expression of specific markers. But for example, supposed to be higher in GCB or ABC in specific subtypes, either subtypes defined by the cell of origin or subtypes defined by the tumor microenvironment sometimes. And we can see additional things that are interesting. We can look at the expression levels of PD-L1, a checkpoint molecule, which tends to be higher on the tumor cells of a cytotoxic cluster, as you would expect, which maybe tells you that targeting PD-1 or PD-L1, in this particular subtype, may be of interest. Some of you may know immune therapy checkpoint inhibition blockade has not done very well in DLBCL, but we think that maybe it's because we haven't really focused on the right subtype of tumors, and maybe I think a TME-based subtyping may actually allow to define patients who may be more likely to respond. The cold tumors, tend to express more BCL2, as you would expect. They actually have less HLA. We can also look at cell size, as I was telling you. So there are also some interesting trends, when it comes to the tumor cells. Now, what's really nice is that we can combine markers both from the microenvironment, from the tumor cells, and we can combine those with clinical factors such as stage or age, and try as hard as possible to predict response to chemotherapy. This is actually done using a machine learning method. This is kind of trying to combine those different features, not in a simple way, like kind of using more like a tree-based structure. And what you can see here on the left side, this is applying the classifier to a test set that did not participate in the training, but we have a pretty amazing ability to separate responders from non-responders to chemotherapy based on the combination of lots and lots of factors, both coming from the tumor cells or from the microenvironment. You see here on the right side, these are the features of the classifier that matters the most. So stage and age, LDH, that matters quite a bit. But for example, CD20 expression on the tumor cells only, that actually is connected to better survival, as you would expect. This is one of the targets or one of the components of the chemotherapy. But looking at blood vessels, right, the structure of blood vessels, that also matters in terms of response. The subtypes and kind of the markers connected to the subtypes I was telling you about, that also matters in terms of response. So the combination of those markers is really what allows us to predict response to chemotherapy in a very efficient way. Another question that we can ask, and I think this is also an interesting part of a talk and something that we want to address more moving forward, is how do these somatic mutations correlate with the tumor cell and the non-tumor cell phenotypes measured using this technique? One thing that we saw, and again, I think we're introducing a new classification of lymphomas, based on the microenvironment. So we do want to understand how it's connected to other classifications, and I told you about the classification based on the ABC GCB patterns. Well, there's actually another classification that's been introduced by the Stout group that is actually based on mutations only. It's this EZB, MCD, and A53, and so on, the classification. Likewise, to our surprise, we actually do not see a very, very strong correlation between our TME-based classification and this mutation-based classification. There are some trends that you see here. For example, the cytotoxic TME is a bit more enhanced or enriched in MCD subtypes. The cold tumors tend to be a bit more enriched in EZB MYC subtypes according to mutations, but these are pretty mild trends. But we do see some pretty interesting correlations. For example, if we focus on the tumor cells, we can see that tumors that have a B2M mutation, B2M is a frequent mutation that is involved in the MHC complex. it's involved in immune evasion of the tumor cells. But what we see clearly here, because we had a marker for HLA that we can look at specifically in the context of the tumor cells, is that tumors that have a mutation in B2M, if you look at HLA MHC1 levels, the tumor cells express much less MHC1 compared to the tumor cells that don't have the mutation in B2M. So that's great, because if you didn't really know anything about B2M as a mutation, this would actually give you some clues about what B2M is doing. And this is also the nice thing about being able to look at specifically the tumor cells. By the way, so what's interesting is that there are some mutations, like you see here, tumors with certain mutations that actually don't have a particularly low levels of MHC1. And so, in all likelihood, this means that this kind of mutation here is actually not an inactivating mutation. That maybe is a mutation that in fact is actually not doing anything to the protein and not affecting MHC. That's also, by the way, a way to credential mutations, right? Specific mutations, whether they actually have the effect that we think that they have. Likewise, tumors with EZH2 mutations. If you look at K27 trimethylation within the tumor cells, you see a pretty high level of K27 trimethylation, as you would expect. And that's, again, only looking at the tumor cells. And what we can do, in fact, is we essentially build a map, right, of the correlation between the presence or absence of a mutation and the increase or decrease of some of the populations of cells that we've seen within each of the tumors. That's the beauty of having data on lots of cases, some of which are going to have mutations and many of which won't have a mutation, and have all these sort of analysis of the microenvironment in the tumor cells. We can actually see quite a few correlations. And I think that's amazing for us because it really tells us potentially what the function of specific mutation is. By looking at this map, you can get a clue about what the mutations are doing, both in terms of the tumor cells. If you have a T here, that's a tumor cell population. If you have a B, that's a tumor cell population. So you see, for example, there are specific mutations like in the BCR, maybe in CPS1 that tend to increase the abundance of this particular tumor cell population. So that just gives you a clue about the function of these mutations, right? I mean, as you know, it's hard to assess the function of mutations, but I think this kind of technique here across so many patients does give you many, many hypotheses that we can use to further analyze these mutations. Another aspect that we are looking at now, and this is very much an unfinished story, it's really looking at spatial compartments. We'd love to be able to assess automatically the presence of compartments within tumors. I think there's an unmet need in that sense. I think there's probably much more structure than we think. We've kind of looked at this, I think maybe we need to look at this more, but we do see when we look at proximity between cell types that we count and assess, that there are some cell types that tend to be closer to each other, maybe because they engage in crosstalks and certain types of communication. In fact, these patterns of subtypes of cells kind of proximity is a little bit dependent on the actual subtype. I think that's something that emerges, really interestingly, and we are still trying to understand what that means. But I think that's, potentially really interesting. This is an area where we are doing some work. This is actually not a B-cell lymphoma project, but we just published a paper looking at the ability to identify compartments within those images that I showed you so far, using a combination of the different cell types, unique markers that they express, and a graph of proximity between cells. We combine those two types of information as a way to find compartments within images that have essentially a similar representation of different cells, and that are close to each other. This technique here works really well on normal tissue where we have annotations that we're able to make ourselves. For example, lung tissue, there's a beautiful architecture of normal lung tissue, and this technique here is able to recover these compartments really nicely with minimal manual intervention, as you can see here. But it also works in tumors, actually, in the bladder cancer data set that we also have here. We've been able to identify different compartments, tumor compartments with immune infiltration, tumor stroma interfaces. This is all done automatically, and we're able to find those compartments. In fact, in breast cancer, we also have a large data set that's published by the bottom middle group, and we've been able to see that this kind of compartment structure, the size of a compartment, of certain compartments at least, is correlated with survival. Like for example, in this picture here in the middle, we can divide tumors based on the abundance of two specific compartments that are stromal rich with lots of T cells versus stroma versus few T cells, and we see a very strong difference in terms of the survival of these patients. So these compartments do tell you something about response to treatment, and I think there's probably a lot more to learn. We haven't applied yet this technique to the B-cell lymphomas, but this is something that we're going to do very soon. And I think that's it. This is the end of the talk. I will look forward to addressing any questions that you would have.
Thank you very much. Olivier.
Thank you.
So,
Thank you.
Michael.
Okay, we'll start. Thank you, Olivier, for your maybe you can unmute your mic. So I have one first question, maybe to start, and then we'll have a couple of questions from the audience, and please ask your question on the question panel or the chat panel. So my first question will be one of the limit, in fact, of this kind of analysis is a segmentation. It's not maybe a limit, but is the tough part of the story. So how do you manage, in fact, this step, in particular using adjacent slide? Because if I understood well, in fact, you have a panel that is integrating. You have two panels, in fact. So you run one panel per slide that are adjacent. So how do you manage that in terms of coordinate? And are you using only homebrew panels, homebrew pipelines on R or Python, or are you also considering commercial tools? So maybe in short, if you have some advice on this particular state. Thank you.
Yeah, no, thank you so much. That's a wonderful question and a very important question. So you're right that the segmentation, I think, is still difficult. The different choices. I think we found some combinations of tools that seem to work well. It's interesting because I have different students using different tools. We typically don't really use kind of commercial tools. We're basically kind of using open source software to do the segmentation. I think what's worked well in some ways, in some settings, is basically having students label multiple cells and essentially teach some of these programs how to do the segmentation, kind of a sort of user-driven learning of what cells look like in different diseases. That's actually worked reasonably well, and I think that's still what we're using. You basically do a little bit of teaching of these methods, and eventually you can just run them on the whole data set, and then you find the cells, and that seems to work okay. But I think what we've seen is also that you need to use multiple markers, because the markers that we have, so we have markers of the nucleus, like DNA, markers of the cytoplasm, you have markers of the membrane, and I think the combination of all these different markers kind of highlights really nicely where the cells are. Because if you only focus on the nucleus, right, you can find the nucleus, you can find the center of the cells, but you can't really find the boundaries of the cells. So we really have to use multiple markers to do a segmentation really well. But we by and large use a combination of open source software as well as some homebrewed sort of tools. I should say that you think about segmentation, right? So there's segmentation of cells, but it's also segmentation of tissue. And so what I talked about towards the end of the talk was much more segmentation of tissue into compartments, right? Groups of cells that kind of form a particular compartment. That's another level of segmentation. But I think what's interesting, some of my students actually looking at doing the segmentation directly from the pixels, because they realize it's actually, if you do segmentation of tissue based on the cells, you're actually missing a lot of extracellular components, like the extracellular matrix, which is not within cells, it's basically outside of cells. And so you want to capture that as well. And so working directly from the pixels to do this kind of high level segmentation, finding pockets, that seems to work actually also pretty well. So I don't know if I answered your question, but I think that's approximately sort of what we're doing now. And so we're not really using commercial software. We basically have our own homebrew sort of type of tools. And so the last part of the talk about the compartment detection. So that paper actually, is something that we put on GitHub, and it just got published. But that's also software that anybody can use, and we'll be happy to assist. If there's anybody interested in using it.
Okay, there are questions from the audience. I have a question from Sylvain Lamure, and the 2 punches are made out from a single surgical biopsy or from different biopsies made at different time points in patients' history?
Yeah, that's a wonderful question too, right? So, actually I get this question a lot, right? I mean, wouldn't it be nice to be able to have multiple biopsies, at different time points, to be able to assess, how maybe the microenvironment is changing, right? I'm not sure we know well whether chemotherapy is changing the microenvironment, for example, but that's pretty fundamental, right? In terms of understanding the mode of action of chemotherapy. As I was telling you, right, I think there seems to be this kind of synergy between the microenvironment and the tumor cell when it comes to chemotherapy effect. But it would be amazing to be able to understand how the microenvironment is changing, as a result of chemotherapy treatment. And unfortunately, we actually do not have, this kind of multi-time point biopsies. We have it in some settings, like in follicular lymphoma. We have many more cohorts. The one I showed you is the one that's a bit more advanced in terms of analysis. But we actually have a nice follicular lymphoma cohort, similar kind of structure, but for some of these patients, we have also a biopsy at a time of transformation into diffuse large B-cell lymphoma. So we have 2 time points, and it kind of is transformation into almost a different disease. But at least there we have 2 time points, and we should be able to understand maybe at least some aspect of the transformation of the microenvironment, when this transformation occurs. But the answer to your question, long story short, is no, and I'm sad because that would be amazing data to have.
Thanks.
Thank you. So another question, one from Suman. Any comment regarding in fact, it's 2 questions. The first one, any comment on cytotoxic lymphoma TME? Why this group of patients are the worst responder? And there are another question. I saw another question from Jerome. Could you identify difference in terms of proliferation or replicative stress in the LBCL cells from cytotoxic T cells and cold tumor groups?
Yeah, wonderful questions. I wish I had probably a better answer than what I'm going to give you. I don't really know, what these cytotoxic lymphoma, why do they respond so poorly to chemotherapy? I don't really understand. I think that the only thing I can say is that I think that the chemotherapy, relies on a semi-functional microenvironment. And if you have an immunosuppressive microenvironment, with lots of bad T cells, exhausted T cells, and lots of M2-like macrophages, which is basically what we see, you just don't get a level of response that, is good. Like what exactly that happens, I'm not sure. The one thing I know, I think I hypothesize, is that if you were able to kind of, modulate the microenvironment to make it a bit more like the more normal-ish microenvironments that we see in other subtypes that seem to respond well to chemotherapy, I think we can sensitize some of these tumors to chemotherapy. So I think, maybe the way to treat those patients is to, maybe give them, anti-PD-1 or PD-L1 inhibitors, and then hit them with chemotherapy. Or maybe, modify the macrophages, right? There are some really interesting, anti-macrophage, or let's say, agents that can modulate macrophage, transform M2 macrophages into M1. I think that, this is also, potentially very, very interesting as a direction. Now, again, why exactly, these tumors don't do so well, I just don't know at this stage. And so, Jerome, you have a question?
Yes. There is a question by...
Oh yeah, so we certainly have proliferation data. I think it was somewhere in the I think there are some the cold tumors proliferate much more. They express more Ki67 in the tumor cells compared to the cytotoxic ones. Replication stress, I don't know that we can tell. I don't know if we have good markers for this in the current panel. But that's a very interesting question. I'll look. I mean, that's something maybe we're able to assess using bulk RNA-seq maybe, but I think that'd be interesting.
There is a provocative statement by Pierre Jobert, and at the end, LDH stage and age are the most important predictors of survival.
Absolutely. I think that's and that is what it is. I mean, but the reality though is that if you really want to improve your ability to kind of distinguish survivals or responders versus non-responders, you need to include additional markers. But you're right, absolutely. ADH, age, these are very important markers. Yeah, honestly, I think age is maybe for different reasons, right? Is that if you're older, I think the ability to tolerate chemotherapy, I think is maybe not as much, and I think that people may die or not do well because they can't really withstand the amount of chemotherapy that is required to be able to effectively treat those cancers, right? So age is important when it comes to survival, but I think it may also be simply because of the frailty of the patients and the fact that they can't they just can't tolerate chemotherapy in the same way that younger individuals can.
I have a naive question. You predict 5 distinct microenvironments with different outcome in patients treated by R-CHOP and on TMA. So how do you manage the heterogeneity and the fact that TMA represent some partial aspect of the tumor? So this is highly restricted. So how do you manage? Is there a kind of normalization of the samples? Did you use many samples for each patient? So how do you manage that?
It is a wonderful question, as the other questions that you asked. I think that unfortunately, so the only thing I can say is that in many patients, and I've been pushing my student to kind of produce a better figure, because I think that's an important figure. I think he has it, but I don't think I was able to include it. But I think that when we have 2 punches per patient, we see that in 85% of the cases, if you are able to put each of a punch within a particular subtype, in 85% of the cases, they actually belong to the same subtype or microenvironment, right? So I think there's actually maybe a bit less heterogeneity within let's say tumors than maybe we think at the level of individual locations, right? At the level of individual tumor. I think the heterogeneity may not be as high as maybe what we think. That's a bit biased, because as I said, a pathologist, chose the punches in the first place, right? And a pathologist, will tend to select tumor-rich regions, right? That makes a lot of sense to do that. And so there's a bit of bias, right? It's possible that I think if you were to look outside of those 2 regions, you would see areas with less tumor cells, but with a distinct microenvironment. So my answer is really that I don't really know. I think when we have 2 punches, they tend to be very similar to each other. But whether that really applies to the whole slide, to the whole tissue, I don't really know yet. We're trying to get some data, for individual slides, covering a much larger area to assess this. I think the data is not super mature yet, but I do think that's a very important question. But I think the strength of this project here is really the number of patients, right? Is the fact that you have hundreds of patients, and so I think that you can, find trends that allow you to overcome, the heterogeneity. It's more like the numbers, and I think we see clear patterns emerging from this very large cohort.
Thank you. So Sarah is wondering at the spatial single cell level, are GCB and ABC B cells surrounded by the same TME subtypes?
Say that again. At the single cell level.
Are GCB and ABC B cells surrounded by the same subtypes from the TME?
So I think what I showed is that within, so you have, so we're able to assess at a high level whether each of the tumors is ABC or GCB, I think, for most of the tumors. But what we do see clearly is that, there is there's much more heterogeneity. So within an ABC tumor, assessed using, some of the IHC mechanisms, for example, or maybe even nanostring, we do see that maybe there are more ABC populations of cells, but there's almost always some GCB cell populations as well, but maybe in a minority, right? But every tumor is in fact a combination. Does that answer your question?
Oh, yes, thank you. Yes, thank you.
I hope it does. If it does not, then I'm happy to. Okay, but no, good. Sarah, do you want to type your question?
Are there other questions?
I think Sarah is typing a question.
Yeah.
So meantime, you can have from Jean. Do you have any information or any idea on correlation with spatial RNA-seq?
No, we don't. I think that what I always say is, spatial RNA-seq is great. I think it's great because you can interrogate many more genes, right, at the same time. But you also lose the resolution, right? The resolution of spatial RNA-seq now is maybe about 20-25 cells. So it's like little neighborhoods of cells, and to me that's I don't know, not very satisfying. Let's put it this way, because then you have to do a deconvolution within each of the spots and count sort of estimate the abundance of the different cell populations. It's not very satisfying in my view. Once you really get down to the resolution of single cells, I think you really get addicted to it, because you basically can see the cells, right? And I think it's just amazing. And the beauty also of this spatial proteomics technique that we use is that it's proteomics. It's like you're measuring things that matter within cells, right? The RNA they matter in some ways, they code for proteins, but they're not really doing the work directly. So it's really amazing to be able to really visualize the protein levels, and so that's why as I say, there's a trade-off between number of genes and the resolution. We tend to like the proteomics method because of the improved resolution. We will see what comes out of spatial transcriptomics on similar samples. One nice thing about also this kind of spatial proteomics technique is that it really works very well with FFP samples, paraffin embedded tissue. It's actually when you've done some genomics like us for many years, you kind of assume that FFP samples are not great for anything genomics or profiling. But it turns out this technique here actually works much better on FFP samples compared to frozen samples, for example. So, suddenly there's all these pathology archives, that we can assess, that are like a treasure troves, it's gold, that we can certainly assess. All right, so let me see. So Sarah asked a question. So, are there different types of different T cells, macrophage?
Yes, meantime.
Yeah, surrounding ABC or GCB cells. That's a great question, Sarah. I do not know the answer. We will look at this, the extent to which around GCB and ABC cells you have different types of immune cells. Wonderful question. I do not know the answer. Sorry. The dialogue between the TME and different types of B cells, you are right on. That says what we need to understand, right? The crosstalks between, these cell populations, like, as much as we do this kind of spatial analysis, right? And you can say, well, at the end of the day, you can just counting the number of cells, right? And that's not great. I kind of started telling you a little bit about the more spatial architecture analysis we're doing, but still not so great. What we need to assess is exactly what you're asking. Are these cells that are close to each other, are they actually talking to each other? Are they engaging in crosstalks? Like, we need to figure this out. I think with this kind of technique, we can figure this out. We can look at ligand and receptors. We need to design a panel basically to be able to assess this kind of communication. So we can assess ligand receptor expression. We can look at where the ligands and the receptors are localized in the membrane. Are they located at the synapse between the tumor cells and the immune cells? I think we can dissect communication between cells using this technique. We just haven't done it yet. I have one student who's been looking at this, looking at cells that kind of like what I say, cells that are in proximity. If you look at the localization of certain proteins, around in the membrane, do cells that are in proximity kind of relocalize some proteins at the location of the synapse, which will kind of tell you that there is some kind of active connection happening. So had a few students looking at this, but without any very convincing results yet. But I think this is probably the most important question that we need to address, is the actual communication between cells.
Okay. You have another question?
I think there's another question for Suman. Yeah. So we good question. There's a question about from Suman. He's asking whether the imaging data that I showed is comparable or compatible with the cyber-cell profiling of the bulk RNA-seq. The paper I showed at the beginning. We haven't looked at this too much. What I think we looked at it at least briefly is that in that paper they had one immune-rich sort of cluster. And I think we're able to subdivide it into multiple different types of immune richness based on the single-cell data. The cold cluster that they have, we also have it. I think it's in the resolution of the immune-rich clusters that I think we are able to do better because we're able to really count the different types of immune cells. And that's something that's very hard to achieve with a cyber-sort. You don't have the accuracy to really kind of get down to the details of the immune cell populations. And I think that's where we're able to do a bit better.
I have a question. In your opinion, what could be the added value of morphological techniques such as spatial transcriptomics compared to RNA-seq with good algos of deconvolution?
Yeah, I mean that's it's sort of what we're addressing now, right? So personally, I come from a quantitative background, so I should be the guy who loves deconvolution because it's like, okay, you're creating stuff out of nothing, right? You have like your mixture, and then with algorithms you're able to deconvolute it, But actually, I don't like it so much, Honestly, I don't. I mean, I know that these deconvolution methods are not that good. They're not as good as I shouldn't say that in public, but I don't think they're as good as people think they are. And I think part of the reason is not so much technical, it's because we don't always know, the markers that are specific to different cell types. And, we basically need a combination of markers to be able to find the individual cells, right? And so that is impossible to get from bulk RNA-seq. You just don't know, how to sort of use markers that are specific to individual cell types, and so I think you end up with a very imperfect result for deconvolution. That's why I don't like it so much. It's amazing if you only have bulk RNA-seq, right? Because then you could go back and it's like, I don't know, there's all kinds of people doing analysis of H&E slides nowadays using AI. It's amazing, right? Because you have like millions of H&E slides and you can go back and analyze it. But at the end of the day, it's H&E slides, right? I mean, there's two markers, and it's good, but it is limited when it comes to the type of information that it conveys. So I think that So basically, my answer is really that I think that this kind of spatial proteomics is like so much better than deconvolution, but sometimes you only have bulk RNA-seq, and therefore deconvolution matters more in that case.
So we still have 15 minutes. Have room for questions, so don't hesitate. One question from Bertrand. Can you further precise the difference between inflammatory and cytotoxic? They are associated with opposite response.
Yeah. So I think it's just really different immune cell composition. We don't typically have in that cell population in the immune rich. We have I think lots of in the inflammatory sort of cluster. I think we don't have these coenzyme B expressing cells. We have less exhausted T cells. We don't have as many M2 like macrophages. So I think there's more of the kind of functional immune cells like CD8 T cells or CD4 T cells that don't express these other markers that are connected to dysfunction. I think that's basically what we're seeing here. But I think the labels, I think that those labels, I think some of them need maybe a little bit of work. I think that my student was sort of made some choices when it comes to the labeling. I think we need to maybe look into the subtypes in more details in terms of the actual label to make sure that it's as accurate as possible.
And the other question was, when you try to integrate your data, all the data, multimodal data, from genomics, transcriptomics, morphology, etc., clinics. And so you describe a machine learning model. What kind of model do you use in this data integration?
Yeah, that's a wonderful question. I also realized that I forgot to answer another question before about the alignment between subsequent, slides. And we typically, as much as we'd like to do that, by the way, this, we have consecutive, tissue sections, and we love to be able to align them, and we realize that's not super easy. So we're actually not really doing that so much, but I think maybe it's a technical thing that we can address better. But just to address your question, Dr. Bousset, about the multi-omic sort of model. So that model actually was mostly combining data from the imaging with clinical factors. It was actually not really using RNA-seq, bulk RNA-seq. It was using information about which markers are expressed in the tumor cells, like CD20, or which markers are expressed or with the fraction of a number of immune cells of a particular subtype, and so on. So it was restricted to the combination of clinical factors and imaging sort of markers. I mean, it is a multi-variable type of model. So in this particular case, we use a form of machine learning called random forest, or kind of an extension of random forest, which is kind of a tree-based, like decision tree-based, sort of type of model. I don't know. It's one of those things where I'm sure we can use better models, maybe even deep learning methods. But we try to keep it simple to some extent, and that's why we decided to go to use that model. But I agree. I think we should probably look at other things.
So another question is this landscape plastic between different biopsies in the same patients, initial and relapse, for an example? And I will add maybe, did you also check to another pathologies about follicular lymphoma or Hodgkin?
Yeah, great question. So as I said, at some point, we don't have, multiple time points. So I don't know, the plasticity of these subtypes of microenvironment. I really don't know. I don't know the extent to which the chemotherapy is changing the microenvironment. I don't know. I think that's a very important question, and I wish that we would know. I think it just we don't have the right samples for now. But I'm hoping at some point we will have the right samples. And what was the second question?
Did you also analyze follicular lymphoma?
Oh yeah, but great question. So we have a data set. We've done the imaging of follicular lymphoma, which is really interesting because it's actually much more structured in follicular lymphoma, as some of you may know. So I think some of the techniques that I talked about at the end, this kind of compartment discovery algorithm, I think that's going to be very helpful in the follicular lymphoma. We just haven't had a chance to analyze the data in detail. But I think this is, we have 150 follicular lymphoma samples already imaged, more or less with the same panel I showed you already. So I think the data is just, there. As much as we like to, to think that we are good at analysis. The more data you produce, the more analysis you need, and so there's never enough computational biology. That's something that we're supposed to be strong at computational biology, but then you start generating data and. It's like always more data, right? It's you always need more computational biologists. So it's like a battle that you can't really win. So we will analyze that data set next and hope to be able to report hopefully interesting results. For now we don't know, but that data set is really interesting because it has actually multiple time points. It has, at least for some of the cases, follicular lymphoma at diagnosis, and then transformed follicular lymphomas into DLBCL. So that's going to be really cool. I think we have about 25 cases where there's transformation like this. So that's going to be amazing, right? Like nobody really knows, right? Why some follicular lymphomas at some point transform into DLBCLs, right? And hopefully we'll get some clues in the microenvironment. Maybe they're all cytotoxic. Maybe they're all cold. Maybe I don't know. Maybe there is something in the microenvironment that's predictive of transformation. We'll see. But I don't know. I don't know the answer yet.
There are many other questions. Michael, do you want to carry on?
Yes, there are more questions. One question from Sylvain. Are you able to assess differential response to CAR T cells or bispecific therapy according to the TME subgroups, as you did with your shot?
What a fantastic question too, right? I mean, everybody's excited about these, these CAR T cell. So we have a tiny bit of data on that, but it's too few samples. Been pushing my students to kind of look at that data. I think we find some patterns. I think unfortunately, I wish I would remember better because we looked at that a little while ago. I think there is a subtype, and I think maybe that's the it might be it's either the cytotoxic cluster or the cold cluster, which is not very helpful. But one of the subtypes which seems to be a bit more connected with response to CAR T cell. Part of the challenge with CAR T cell is that they're given to patients, after they failed everything else, right? So getting to the bottom of how the chemotherapy is changing the microenvironment is pretty essential, right? Because otherwise we could if you don't kind of address that, then you could think that CAR T cells will work better in the context of one microenvironment, but it's the result of essentially the modification of the microenvironment by the chemotherapy potentially. So, but that's a very important question. We have some data, but a few patients, so the results were not very conclusive. But I hope that at some point we can readdress this question using a larger cohort.
There is a question by Bertrand. Fascinated that there is no obvious correlation between genetics, ABC, GCB, and TME. Can you comment?
Well, I think this is the fact that there is minimal correlation between the mutation data, somatic mutation data, all of the cell of origin. I think it's probably the most puzzling, kind of sort of finding, I think, in my view, of this whole project. Because you have 5 different types of microenvironment, right? But what creates them, right? Clearly we're not finding any correlation with mutations. So it's not the fact that the tumor cell acquires a mutation that therefore, then it's changing the microenvironment, reprogramming the microenvironment to kind of fall within one of the 5 subtypes. There's no clear correlation. So what exactly is happening, right? So I don't know the answer. I think one possibility is that tumor cells establish themselves non-randomly in a particular location, maybe in the lymph node or a particular place, and kind of is co-opting the existing microenvironment, right? And kind of thriving in this location. It doesn't matter so much, what the mutation status of the tumors is like. The tumors is like, it's all about the soil. It's like they establish themselves and they grow and they basically kind of inherit, if you want, a microenvironment of some sort. So I think that's a possibility. I mean, another possibility is that, we've been looking at individual mutations, and maybe it's a combination of mutations that matters when it comes to, dictating the microenvironment. I don't know. But the fact that we don't see a clear correlation between mutations, we see some trends, right? But not very strong correlations. I was expecting some very strong correlations. And we didn't find those. And again, as I said, I can only hypothesize whether that's the case, but I think it's a really important question.
We have time for a few questions. Michael, do you want to select that?
From Andrew. Regarding the segmentation, do you have any orthogonal data to validate the quality or accuracy of segmentation?
So I'm assuming that you're talking about segmentation of cells, right? So there's multiple, as I said, there's multiple layers of segmentation, right? Segmentation of cells, and then there's segmentation of tissue into kind of neighborhoods, right? Or let's say little pockets, and then there's segmentation into compartments, right? So, the paper that just came out from our lab, a couple of days ago, is looking at segmentation into compartments. And so for that, we had like normal tissues, like I said, lung tissues, annotated with a pathologist who essentially drew on the actual, slides, the different structures. And so we were able to try to assess the ability of our unsupervised method to recover those different compartments, and it was very good. But when it comes to segmentation of cells, we early on, I think we let's say we kind of decided to focus on this kind of supervised methods, where we train a classifier with enough annotated cells, and we kind of let the classifier find the rest of the cells. That seems to work really well. I would say that we did some kind of visual analysis, maybe not a systematic assessment of the quality of the segmentation of cells, but I think that overall we've convinced ourselves, by looking at enough images, that the quality was good. But maybe that's something we can revisit. I think it's as we discussed at the beginning. I think Dr. Roussel, you mentioned that. I think that's a very important step, right? It's like the first step of the analysis. So if you mess it up, then everything else is messed up. But I think we have a good handle on that. And I'd be happy to put you in touch with some of my students, and they can tell you more detail. What are the steps, exactly the steps that they used, to achieve segmentation, and maybe can give you a better sense of quality.
So you have a lot of success, and there are many questions, and we will not have enough time to address all the questions. So I'm going to select one or two in a row. First question about PD-L1. What about the expression of PD-L1 by tumor cells or by the macrophages according to the cell of origin or the TME signature? By Jean-Philippe Mervieux.
Yeah. So I think I showed a plot really quickly. So we'd see a lot of PD-L1 on the tumor cells in the cytotoxic cluster. They're still in our range, but I think on average, we tend to see more PD-L1 on the tumor cells in that cluster, which is why I was saying, it's very much possible that this is where the more likely responders to anti-PD-1 or anti-PD-L1 will be located. As I said, the PD-1 treatment has not been very successful, actually has failed in diffuse large B-cell lymphomas. And I think maybe partly is because, we kind of give it to everyone, right? And I think that there's probably a small subtype of patients who may be more likely to respond. These are the PD-L1 expressing sort of the cytotoxic, what we call cytotoxic, and clearly the tumor cell seems to express more PD-L1. Now, we may have looked at macrophages with PD-L1, but I don't remember the results. We certainly have that data because, we know where the macrophages are, and we stain for PD-L1, so we have that data somewhere. I don't know that we looked at, but I think we should look at it.
There is a question by Philippe Gola. Is this B-cell heterogeneity, ABC, GCB, in every tumor with the neoplastic cells, or does it relate to the presence of reactive cells in the TME? In addition, what about this approach on core needle biopsies? Have you access to this kind of material?
Yeah.
It's a long question.
Fantastic question. So, we didn't really, I think that's something we need to look at, right? The extent to which, around the ABC or GCB you have, different types of immune cells. We haven't looked at that so much. In fact, we haven't looked at that at all. But I think that's a very important question, and we should really look at it. The needle biopsy, so I think that's not a problem, right? We can, so basically the tissue microarrays is basically kind of, cores, right, that you take, out of blocks, from larger samples. And so, you don't need a lot of cells, right? You can take a needle biopsy, kind of, cut it through, take a slice of it, and image it, so you can absolutely do this kind of technique using needle biopsies without any doubt, and it works really well. We just haven't done that, in this particular case.
Okay, there are other questions, so we have time for one or two last questions because you have a lot of success, and you talk subject of hard debate.
It's funny because I can't see the questions, so I believe you. I saw some questions at some point, and then there's
if you catch one, if you're interested in you can address it.
Okay. They don't show up anymore on my list of questions. My chat is, there were some questions there, but I don't see the other questions. Do feel free to send me an email. What I'll do is maybe I'll put my email in the chat so that if anybody has pressing questions that you really want to know the answer of, do feel free to send me an email, and I'll be very happy to answer.
Mohammed Halal has a question. Hello. How would you define the geographical extent of tumor microenvironment on the slide? He asked, what would be the borders of the TMA in the DLBCLs?
Yeah, so I'm not sure I fully understand the question, or let's say I'm not sure my interpretation of the question is right, but so obviously there's probably some patterns of architecture within those those cores. We try to look at it.
But the infiltration front of the tumor. I understand.
Oh, I see what you mean. Yeah, okay. So that's that's a very good question. That depends on the core, the cores that were selected. And as I was telling you, I think those tend to be selected by pathologists, who because of the choices that they make, will focus on the areas of high cellularity, and maybe will focus less on the invasive sort of margins. But you're absolutely right. I think that would be really fascinating to kind of better understand the margins between the tumors and the rest of the tissue. And we haven't done that much so far.
Okay. Great.
So we have to conclude.
Yeah.
We are more than in time. So thank you very much. There are some remaining questions, but we'll send them to you, and you will be able to respond to one or two additional questions. So thank you again very much for your amazing talk, your presentation of massive data. That was very, very impressive. Thank you. And thank you.
Thank you.